Hello, it is time to talk Tully. These are Tully monsters up here. I couldn't find one that's moving around, but you're just gonna have to imagine them moving around and going about their daily lives. So, Tully monster is probably, to get this out of the way at the front, something like a highly modified lamprey. And if lamprey doesn't mean a lot to you, here's a lamprey. So lamprey are these things that are going to attach onto other fish. They're going to suck their blood, basically, like little vampires of the ocean. And so they have these little teeth here, but they don't have a jaw. The teeth are just there as a way to hook into and tap into the other animal that they are going to feed on. You'll notice the lovely little sort of gill slits down the side. They look like little holes, and the nice big eyes that we have there. So that's a lamprey. Those still exist. Those are fine. Here is a carboniferous lamprey. So now we're getting toward the right time. The Tully monster, we should point out, is the state fossil of Illinois and is from Mazon Creek. Yeah, it's only known from there as far as I'm aware. And that's there are not many of them, but they're really cool. So this is more of a traditional, normal-looking lamprey. So just compare these two things for a minute. All right, you got this kind of diagonal set of gill slits, and here they are on the side. You got the large eyes. Here are the eyes, but they're extended on what they call the bar organ. And I don't really know why this is a thing, but I don't know why hammerhead sharks are a thing. Yeah, they're presumably good at their job, and I'm not as much of an ichthyologist as maybe I ought to be. And then look at the mouth. Instead of having the mouth just truncate right here, it's got this extended sort of tube-like mouth, and still the same teeth there without really having a jaw. It's just teeth that are together that are, I don't know, yeah, kind of serving the same purpose as a jaw might serve with teeth in it. All right, so that out of the way, let's dig into the actual Tully monster fossils. And here, we don't need this guy anymore. Let's get rid of that. So this is the latest, I believe. There was one article after this, but it didn't have a ton of things to say about this positioning, if I remember correctly. So the idea is that this is going to be related. Here's Tullimonstrum. Here's Maomizon. That's the that's this guy, little lamprey from the Mazon Creek formation. And down here you can see it's related to Euconodonta. That's the conodont animal. Remember that thing with all the little itty bitty teeth that everybody uses for biostratigraphy? Yeah, that thing. Yeah, where we only have a few copies of the actual body with the teeth in place. Yeah, it's a big bug-eyed, jawless fish creature with an amazing array of teeth down inside of it. And then they're saying that this is how it relates to other fish of the world, other sort of fossil fish for the most part, and some other groups of fish that are around. And so that's reasonable. Look inside of this, you've got a gut that's coming through here, maybe something like a crop up here. The actual brain would be attached to the eyes, so it'd be all the way back here. So this is like a trunk, this sort of proboscis. And then it's going to have its gills that are seated behind the eyes, so there we are. And a tail and nice little muscle blocks for flopping this tail around and presumably swimming through the water. So we're thinking this is some kind of predator, although maybe not like a big-time hunting predator. Maybe it's going after small things. It's really unclear what exactly Tullimonstrum's place was in the world. Yeah, at least from these few papers. So, this is looking at Tully monsters. And so this is from the paper where they come out, and for the first time in this paper, they say they're actually probably vertebrate animals, which is what we're implying with all this other stuff. These are all vertebrate animals. They're just weird vertebrates in that we don't, see them every day, and they don't look like us. But yeah, they fit perfectly well on the tree. Sort of at the early branching end of where some of the fish are. And they're saying that, all right, what do we see in Tullimonster? So they're saying, well, we don't see. So otic capsules are like the capsule around the eye. We're saying we don't get that preserved, but in other animals it is preserved. And so they're saying these other animals, which are from the same site, preserve this feature, but Tully monsters don't. And so they're saying, so maybe this wasn't there. That's the idea. They're saying the Tully monster preserves a notochord. At least that's what one group contends. The other group contends that we can't really be sure that that's what it is, but it could be. And they're saying that all these other things, which definitely would have had a notochord, as far as we're aware, they should be at least chordate animals and possibly vertebrate animals. That is not preserved in this site. Myomeres are muscle blocks, and you do get those in some papers, two different papers about Tully monster. And so on. You get a nice gut trace in there, even when you don't get gut traces in some of the other animals from the same site. So something was a little different about either Tully monster itself or the way in which it was fossilizing, and it is difficult to sort that out, but everything I'm going to tell you about Tully monster is not without problems. There are definite problems here, just uncertainties that can probably be solved with just a few more sites with extraordinary soft body preservation of fossils from around this time. I mean, is that too much to ask? Surely not. Yeah. In a way, we kind of need more mining. A lot of mines are where these amazing fossils come from. I don't know. That's not, yeah, that's not great on other levels. But for the paleontology, yeah, maybe worth it. So here are some of the things that they are comparing Tully monster to. They're saying, hey, it looks a little bit like a tunicate larva. It looks a little bit like something like a lamprey. It looks a little bit like these little things, cephalochordates. One of the famous ones is called branchiostoma. And these are really, really primitive chordates, just right there, super early branch. Vetulicolia, which we've covered elsewhere, are from the mid-Cambrian through the end of the Cambrian, I think. And these have been called, they've been said to have at least some chordate-like traits. And you can see the gill openings on the side, which actually resemble those on here, but it's probably unwise on an animal as weird as this to get really bogged down in any one of these features that could be sort of convergent evolution derived. Yeah. All right. So here are some of the features that we see. So myomeres, these are those muscle blocks. This is like what you see inside of cooked fish, just where it sort of goes into chunks in kind of columns of muscle. And as a body of a creature or a fish or whatever dries out, this is roughly what you would see. And so this is probably happening during the fossilization process, after death, possibly after some of the concretion has started to form around this, because Tully monsters are only known from those nice siderite concretions. And then they measure the body length, and they show, okay, this thing grows, and the number of segments in the muscle stays fairly constant. It may slightly increase with increasing size, but that's pretty close to a flat line, which means that they're not undergoing a metamorphosis. They're not developing more muscles. It's just their muscles are growing as the body grows, which is kind of a nice chordate thing. There are some other invertebrates that undergo horrendous, actually some vertebrates too, just amazing metamorphoses even as adults, but that's for another class. All right. Here is a picture of the brain of a Tully monster. And it is really faint. And here you can see two examples. You've got this sort of super faint bulb. Here is the bar organ, which is going to connect to the eyes. And you just got the little sort of carbon trace where the head split open right there. And then you got this bulb at the back. So it's kind of like three little bulbs there that make up the trilobed brain. Yeah. And yeah, so this is where it would be in the organism, all the way back here by the eye, essentially. Okay, here's the evidence for a notochord, and see how strong you think this is. These are, there's that little line running through, so maybe. Here is a sort of a shadow. It's a little different color. It's not lining up with the muscles. It is running down right where the spinal cord would be. So I think it's plausible, but I don't think that is definite proof at all, which some other authors have also said that, hey, yeah, you're jumping ahead here. But it seems reasonable, and that's the currently accepted understanding of Tully monsters. And I don't know, ask me in a couple years, we'll see what the new papers say. But that's where we are right now. Here is the proboscis. And on a lot of the specimens, there does seem to be a real kink in it, right about there. And so what you're seeing here is maybe how they carried it in life, or maybe they had it pulled back and they could reach out and try and grab stuff like that. I don't know. At the end of the proboscis, it's sort of folded over, almost as if it was like a jaw, but it doesn't have bones in there. It doesn't have, as far as we can tell, it didn't work like a jaw, really. But there are little teeth in there, so it's got little teeth without having any sort of detectable jawbone inside. And they're comparing this to other kinds of teeth that you see at this site. And so, yeah, little tiny teeth. That's a good sign. Let's see. Here we are looking at it a little bit closer. Here you can see the little teeth, and they're trying to figure out what the teeth actually are. Are these sort of little hollow structures that are going to be sitting on some sort of muscle or connective tissue, or are these things embedded down into the jaw tissue? And they decide they're kind of on the surface, but they're sort of attached from inside. So not really like teeth as you and I know them. Yeah, a different sort of tooth. Here are the eyes. And so remember, they have that bar organ, and the eyes are at the ends of this. And if you zoom in on the eyes, they look black. So, okay, that's fine. Because if they are chordate animals, then they should have melanin making up the back of their eye. And it's going to be in these little melanosomes, which would look something like this. Now, let's see. One of these is a modern animal, and a couple of these are from the Tully monster fossil. Actually, no, those are all Tully monster eyes. That's right. In the paper, they have another figure that shows other animals and what the eye looks like. Yeah, here, that's coming up. All right. Yeah, let's not jump ahead. But yeah, so these little blobs are going to contain the melanin, and they look a lot like the way melanosomes look at the back of a chordate eye. Here are some other fossils from Mazon Creek, and you can see the little melanosomes fossilized. That's pretty crazy. And they have different appearances, but these are all things that are demonstrably fish, that have bones that actually at least left outlines of the bones, and they're very recognizable as fish. So yeah, we're examining the eyes of those. Here we are. And yeah, similar features. So this is perhaps the strongest case. The finding the maybe notochord, maybe not, is certainly, I don't know, it seems like confirmatory evidence at best, if that is what it is supposed to be, and I don't know what else it would be, but you know, it's not the strongest evidence, I would say. But yeah, yeah. So it's pretty neat that you've got this good a preservation in the eyes of these creatures. And they've actually done some other studies where they examined the melanin pigment and showed that it does appear to be to look like fossilized melanin. So that's kind of interesting. And so here they're looking at different eye types. And melanin as a screening pigment appears in a lot of different types of animals. Here they are showing it all up and down the tree. And so that would be an argument to say that, well, maybe this could be anything. This doesn't have to be a chordate. But to have them in cylinder-shaped melanosomes, they're saying, is something that you only get in whatever Tullimonsters are, and all of fish, and all of, things that come from that. So, yeah, presumably this is what we look like as well, with these little cylindrical melanosomes. I don't know. Does that prove it for you, or does that tell you that these are at least allied with primitive chordates, but maybe something else? I think that is a tough call still, but at least we're in the right part. I mean, this is not a cnidarian. I don't think anybody is suggesting that. It's not a flatworm. So we're narrowing it down. Doesn't appear to be an echinoderm. It just really doesn't look like one in any way. So something definitely in the deuterostomes. So something that would share an ancestor with echinoderms and regular chordates. These are the like sharks and bony fish down here. And they're showing two possible positions for that. So maybe, why not? Here is the actual molecular evidence that these are preserved melanin. And so they're showing sort of fresh melanin from samples. And these are from crow feathers. That's Corvus coronae, I believe. Here's the Tullimonstrum melanin. And so they're getting a lot of similar peaks showing up here in the Tullimonstrum melanin. And they mark them with red dots wherever they think the peaks are. And so the pattern is roughly similar. And you got to figure that the melanin has undergone some degree of diagenesis in the time since fossilization. So that probably makes sense. If you take fresh melanin and you heat it up and you add pressure to it, the diagenesis pushes it over toward where you see all of the fossil melanin. From Tullimonsters, I think, are the either the X's or the dots. I don't know. We'll have to look. And the others are from other fossilized certain chordate animals. So here we go. This is comparing Tullimonster eye pigment, melanosome pigment, to Eocene frog melanosome pigment. So here we've got, it looks as though we're doing mass spec on this. Yeah, by the look of it. Oh no, it's actually sim, time of flight sim. So that's a, it's a photoelectron coupled mass spec technique. You get a lot of information about elemental content as well as mass, if I'm remembering that correctly. You're welcome to look that up and Wikipedia yourself to knowledge, because why not? Tullimonster eye is here, and the extant chicken is here, and here's an extant crow, and here is a frog, fossil frog, fairly recent fossil frog. Here is a Jurassic squid, but it's still got melanin in its ink, and you can see very similar patterns here in each of these readouts. So yeah, seems like that is probably melanin. So there you go. We started with controversy, like maybe it's not, maybe it's something else, and we ended up with, I don't know, I guess it's probably a chordate. But those are Tullimonsters, and Tullimonsters are fantastic. Now if you ever see one in the museum, you will know what you're looking at on this long squidgy red rock. I think they're about that big. Yeah. So thank you very much. This has been Tullimonsters.